Welcome to another tutorial with Caps Lock. This is Yasho, and today we're going to continue where we left off discussing the hypothalamic pituitary thyroid axis. In particular, we're going to take a look at some thyroid histology, linking this topic to the TOB module that we have, and we're also going to discuss how T3 and T4 are produced by the thyroid follicles. So just a quick recap from our previous session, we mentioned that the hypothalamus, which is located in the brain, produces TRH or thyrotropin releasing hormone, which then travels or descends down to the anterior pituitary gland to stimulate the release of TSH or thyroid stimulating hormone. TSH then travels through the bloodstream to where the thyroid gland is located in front of the neck, and it binds to cell surface receptors on the gland to stimulate the release of T3 and T4. And this axis is controlled and maintained by negative feedback. Now, if I zoom in on the thyroid gland here, and we're looking at it from a microscopic perspective now, we can see many of these functional units that are responsible for the production of T3 and T4, which are called the thyroid follicles. And these thyroid follicles are often described as a ring of cells surrounding a ball of fluid. Now, the fluid inside the lumen of the thyroid follicles is a thick viscous liquid called colloid, which is actually Latin for glue-like. And the ring of cuboidal cells that surround this structure are called the thyroid follicular cells, which are responsible for secreting T3 and T4. Apart from the thyroid follicles, if we explore the structure of the thyroid gland a bit more, we can discover another type of cells called the thyroid parafollicular cells. These are the cells responsible for producing calcitonin, which is another hormone produced by the thyroid gland. Calcitonin's main function is to oppose the action of PTH, or the parathyroid hormone, but I'm going to talk about it in more depth when we're going through the PTH axis. The point I'd like to emphasize here is that the thyroid gland produces three main hormones: T3, T4, and calcitonin. T3 and T4 are made inside the thyroid follicles and released by the thyroid follicular cells. Their main function is to increase our basal metabolic rate, amongst many other actions, such as increasing our heart rate and gut motility. And calcitonin, which is produced by the parafollicular cells of the thyroid gland, has the function of opposing the action of PTH, or the parathyroid hormone. Okay, so we're still zoomed in on the thyroid gland, and we can see some blood capillaries and red blood cells here. You don't have to know this at this stage, but the thyroid gland has a dual blood supply from the superior and inferior thyroid arteries. So hopefully now we have a good grasp on the histology of thyroid, so let's take a look at some histology slides from our TOB module, which could be simple possible exam questions, and let's see if we can figure out what these red arrows are pointing towards. Okay, so let's go through this slide together. We can see some thyroid follicles here, like we talked about. And inside the thyroid follicles, inside the lumen, they have this thick viscous liquid called colloid. The lumen of the thyroid follicle actually acts as a reservoir or storage for T3 and T4, and it's where these hormones are initially synthesized from. And surrounding the thyroid follicle, as we talked about, we can see the cuboidal thyroid follicular cells. They're also called cuboidal thyrocytes sometimes. These are the cells that secrete T3 and T4. And on the outside of the thyroid follicles, we can see the thyroid parafollicular cells, which produce calcitonin. We can also see some capillaries here, and obviously inside them they have red blood cells, which are visible in this particular slide. Okay, so let's view another slide together, and in this one it's already telling you that this is taken from the thyroid gland. And please do read the information that they give you about the slides and the questions quite thoroughly, because they often tell you the location where the sample was taken from, and from the location you can kind of tell the cell epithelia type, for example, and it makes it much easier to navigate through the slide itself. So again, in this slide we can see the thyroid follicular cells, which release T3 and T4. Another name for them is the cuboidal thyrocyte. As we said, their epithelia is cuboidal. And we can also see some capillaries and red blood cells as well. Okay, I'd like to move on now and talk a little bit about how T3 and T4 are produced. So the two main constituents, or the raw materials if you'd like, that we need to make T3 and T4, the thyroid hormones, are tyrosine and iodine. And you can kind of derive this from their name. T3 stands for triiodothyronine, which means it has 3 iodines attached to tyrosine residues. T4 on the other hand stands for tetraiodothyronine, or thyroxine for short sometimes, and that means it has 4 iodines attached to tyrosine residues, or tyrosine rings. So we've established that we need iodine and tyrosine to be able to make these hormones. The way we get the iodine is from consuming iodide in our diet. And in fact, many of the dietary products that we take, such as table salt, have artificial iodide added to them as an effort to prevent iodine deficiency, which can lead to hypothyroidism. I will discuss this in more detail in the next session. Okay, so we take in and digest this iodide, and it's transported to the thyroid follicular cells through a channel called the NIS, or the sodium iodide symporter. And this is done against the concentration gradient. And once the iodide is inside these thyroid follicular cells which surround the thyroid follicle, it continues to travel to inside the lumen of the thyroid follicle. And this is done through a channel called pendrin. And once this iodide is inside the lumen of the thyroid follicle, it has to be converted, or in better words, oxidized to iodine to be able to attach to tyrosine residues, or these tyrosine rings. And the enzyme that oxidizes iodide to iodine is called thyroperoxidase. And again, this is happening inside the lumen of the thyroid follicle. So now we have our free iodine, which can attach to these tyrosine rings to produce T3 and T4. But where do we get these tyrosine rings from? Well, the thyroid follicular cells produce a protein called thyroglobulin, which is essentially a long chain of these tyrosine residues attached to each other. And they're moved from the thyroid follicular cells to inside the thyroid follicle lumen by exocytosis. And once they're inside the lumen, the free iodine that we have, which was converted from iodide by thyroperoxidase, can attach to these tyrosine residues, which are in these long chains, in 2 ways. You can either have one iodine attaching to one spot on the tyrosine ring on these chains, which produces a unit of MIT, or monoiodothyronine, or you can have 2 free iodines attaching to a tyrosine ring, which produces a unit called DIT, or diiodothyronine. And once these units of DIT and MIT are formed, they can further couple together to produce T3 and T4. So if an MIT unit, for example, couples with a DIT unit, we're going to have MIT which has 1 iodine plus DIT which has 2 iodine, and that's going to give us a molecule with 3 iodines attached to tyrosine rings, or T3, or triiodothyronine. On the other hand, if we have 2 DIT molecules coupling together, so DIT plus DIT, that's 2 plus 2, which is 4, and that's quick maths. I'm going to have to re-record this section. And that's going to give us a molecule with 4 iodines overall attached to tyrosine rings, or tetraiodothyronine, or T4, or thyroxine for short. The point that I have to emphasize on here is that when these molecules are in the lumen of the thyroid follicle, in this colloid that we talked about, they exist as these long chains of T3s and T4s connected to each other, and they haven't been separated or cleaved from each other yet. When TSH, or thyroid stimulating hormone, which is produced by the anterior pituitary gland, binds to cell surface receptors on the thyroid gland, it kind of stimulates or sends a signal to the thyroid follicles to take these long chains of T3s and T4s, which are stored inside the lumen, to be taken back into the thyroid follicular cells through endocytosis. These long chains of T3s and T4s are then cleaved or separated from each other when they're inside the thyroid follicular cells using lysosomal enzymes. And once they're separated and cleaved from each other, we end up with these individual entities of T3 and T4, which are then released through the thyroid follicular cells. A final point that I'd like to mention is that once T3 and T4 are in the bloodstream, we have a protein called TBG, or thyroid binding globulin, which is produced by the liver, and it binds to T3 and T4 and acts as a taxi and effectively carries them to the targeted organ. I hope this tutorial was helpful. I realize it was a lot of content in a very short period of time. If you do have any questions for me, please do get in touch with myself or anyone else from the CAPSOC team, and we will be more than happy to help. Thank you so much. In the next tutorial, we will go through hyper and hypothyroidism and some of the conditions that could arise from thyroid gland dysfunction.